enzyme-substrate complex is a combination of a substrate entering the catalyst. The substrate fits to the active site of the enzyme and attaches perfectly. Each substrate perfectly fits into the active site of the enzyme, like a key is specifically designed for its lock. Oftentimes, a substrate does not match the catalyst, and a reaction does not take place. Inside the complex, a substrate molecule undergoes a chemical reaction and is transformed into a new product.
Competitive inhibition occurs when substrates compete to bind to an enzyme. An inhibiting substrate binds to the active site of an enzyme first and prevents others from entering the catalyst. As a result, only one of the substrates can undergo a chemical reaction.
Non-competitive inhibition is evident when one substrate binds to an enzyme at a place other than the active site. As a result of this, the active site is changed so that another substrate is unable to bind to the active site. Thus, the substrate shapes the enzyme so that other cannot undergo a chemical reaction. The inhibiting reaction forbids other reactions.
Allosteric regulation is very similar to non-competitive inhibition. However, the entrance of a substrate into a place other than the active site changes the shape of the enzyme for the benefit of other substrates. A substrate enters a catalyst and forces its whole form to change as a result. However, a substrate that would not fit into the active site before is now able to enter into this altered site, known as the allosteric site. Thus, the change in the enzyme helps another substrate undergo a reaction.
Feedback inhibition occurs when the active site in an enzyme is full, and one of the substrates inside the enzyme leaves temporarily to instruct the other substrates not to enter. The enzyme reaches a critical concentration and could be in danger if another substrate enters the active site. So, one of the substrate inhibits others on the outside in order to protect the chemical reactions.
Ana means family.
An enzyme's homeostasis is regulated by two factors, pH and temperature. If the enzyme is in a condition that threatens its stability, it can damage. This is why proteins must be in careful condition so chemical reactions can occur.
You cursed rat! Look what you've done!